Today we're going to dissect the squid, and the first thing we want to do is orient ourselves. This is going to be the dorsal aspect, and the head end, where the tentacles and the arms are located, this is going to be the ventral aspect. After we identify those aspects or regions, let's go ahead and make sure that the siphon side is up. At this point, we need to identify the external features. So we've got the fins. There's one fin. Here's another fin. And the squid's going to use these fins for steering and also for staying upright. We can see this very muscular mantle. And the squid is going to use this muscular mantle to, one, for protection, and two, the squid is actually going to pull water inside this muscular mantle. And inside the muscular mantle are its organs. But the squid is actually going to squeeze the water after it pulls it inside, and that water is going to exit through the siphon. You can see that siphon right here, but the water, when the squid squeezes this mantle, that mantle literally seals around the siphon. And the only way the water can exit the squid is through this siphon, and it exits it just like a garden hose. The water goes out one direction, and the squid goes the opposite direction. Newton's third law of motion. For every action, water going in one direction, there's an equal and opposite reaction. Squid moves in the opposite direction. And the squid can move very effectively, about 25 miles an hour through the body. In fact, some people have actually described them as little missiles going through the water because they're so fast and they're so aerodynamic. If we look at the side of the head, we can begin to see the eye. And of course, the proteins have been denatured, so they're a little bit discolored. And we can see the eye on the other side. We can identify it. There it is. And we can actually feel that. We can feel the difference. You can actually feel that eye. So again, that's one of the things I want you to do when you're dissecting organisms, is feel structures. It's not about just identifying structures. It's about being able to feel what an eye feels like, feel what cartilage, what bone feels like, feel the difference between an artery and a vein, feel what glandular tissue feels like, being able to identify a nerve both by sight and by touch. So that's another thing you want to get out of dissection. And of course, you want to be able to reveal the various structures without destroying them in the process. So all those things are going to be part of dissection. Now, moving on with the squid, we can see all the arms. So the arms are going to be these shorter structures, and the two very long structures are going to be the tentacles. The tentacles are used to initially grasp the food, and then the arms eventually help out. That food is then brought in, and if we kind of spread things apart here, we can see the mouth. And if we zoom in on the mouth, you can see that hole. And inside that hole, and we're going to see this in just a few minutes, there is a beak. And the beak almost acts like a chisel, and the squid brings that food in very quickly using its arms and tentacles, and it rips that food right across that beak or chisel. And then it puts that food in its mouth and grinds it up into small pieces. So make sure you identify all those structures. Now, the first thing I want to do is I want to find the eye of the squid. And then I'm going to take my scissors and I'm going to make just a small incision, just a little snip in the eye. And I'm going to go ahead and enter the eye. I'm going to use the probe and I'm going to scoop around a little bit. And I'm going to look for, and it just flew out, I'm going to look for a small hard pebble. I can feel it. The small hard pebble is the lens. And again, that's used to focus the light. And I wanted you to feel what a lens feels like. Now, in real life, this would be clear, this would be soft, but it's been denatured by the preservative. The proteins in there have been denatured. So it's just a hard pebble now. But make sure you identify that. And then when we do the cow eye dissection, that lens is going to be much larger, about the size of a small gumball. Try to get that out of one eye, and then go over to the other eye and do the same thing. Make a small incision, kind of scoop around a little bit, and see if you can find that lens. Again, you've got to feel for it periodically. Once you've done that, the next step is we're going to go ahead and open up the squid. We're going to cut the mantle ventral to dorsal. Now what I like to do is I actually like to squeeze the mantle a little bit, kind of tent it up, because underneath this mantle are a lot of soft organs, and I don't want to damage those soft organs. So when I actually cut the mantle, I want to keep this lower blade parallel with the mantle. I don't want to angle it down and gouge up the organs. So let's go ahead, and I'm right above the siphon also. Make sure you're right above the siphon. And then slowly just cut all the way up. Being careful not to damage those internal organs if at all possible. All the way to the end. Now once you've done that, let's go ahead and pin the mantle back. Now when we pin the mantle back, it's going to be important to pin it at a very large angle, like this. Maybe about 30 degrees. That way the pin doesn't pull out of the mat. So I'm going to go ahead and I'm going to pin the other side. And I'm pinning this diagonally across the tray. Move this. Maybe about 4 pins on each side should do the job. One last pin. There we go. A couple things I want you to be able to identify right away. The siphon. If we look at the mantle, you can see this rib-shaped structure here and another rib-shaped structure over here. Those are cartilage plates under there. I want you to feel what that cartilage feels like here and here. Also, if we reflect the head back, you can see this cartilage material. It's visible. I want you to identify the cartilage. I want you to feel the cartilage. This lends the squid additional support. It's part of his skeleton. We can see some retractor muscles. These retractor muscles are going to control the siphon. I'm going to show you those in just a minute. All this blue material is latex. That's been injected into the squid's venous system so that make it a little bit more prominent, a little bit more obvious. If latex is not injected in there, the venous system collapses and we really can't see it very well at all. This structure and this structure over here, those are going to be our gills. As water comes in the mantle, this is the mantle cavity. It's going to bathe those gills where gas exchange takes place. At the base of the gill here, I'm going to free that up, and also over here. And I'm going to free that up a little bit. I like to use a probe for this because it's dull and it doesn't tear structures up. These are referred to as the brachial hearts or the gill hearts. They actually pump the blood to each gill. And then in the center, right here, is going to be the systemic heart. This heart pumps blood to the entire body. It's a 3-chambered heart. Squid needs more efficiency. So it's a 3-chambered heart to pump blood to the muscles and to the other organs. If we continue dorsally, we see all this looks like saran wrap. This is going to be the peritoneum. It surrounds the organs, offers protection, probably against infection. I'm going to reflect back that saran wrap material carefully. Just tease structures apart. There's a structure coming into view right here. This is going to be the vas deferens. This other cream-colored structure is going to be the testes. So this is a male squid. This is the testes. If this were a female, we would see two large nidamental glands, one here, and then we would see the ovary up here further. But this is going to be a male. And this very soft material, kind of pouchy, kind of like a balloon almost. You can see it right there. It's decomposed slightly due to the preservative. This is going to be the cecum. This would be almost like a balloon. It's very thin, and this is where the majority of absorption of nutrients is going to take place. So that's the cecum. That's going to be the cecum. Now, in order to get to the cecum, the food has to be digested previously in a pouch-like structure called the stomach. And I'm looking for that right now. I'm sort of teasing material away very gently. And now, coming into view, you can see this pouch-like structure. That's going to be the stomach. Go ahead and feel that. You can feel that pouch. It's more muscular, a little bit thicker. That's where food's going to get further ground up. And then from there, you can see this very soft, pouchy material coming off of it. That's the cecum. From the stomach, the food's then going to go into the cecum, where the majority of absorption of nutrients is going to take place. Underneath, you can see this pinkish structure. This is going to be the pen. That's the squid's internal shell. And it uses that shell for support. The next thing we want to do is go back towards the siphon. And I want to identify this sort of silvery structure right here. You can see that silvery structure. If we zoom in a little bit, that silvery structure there, it continues on down. This is going to be the ink sac. That's the ink sac. We don't want to puncture or cut the ink sac right now because potentially ink could get all over, and that would be messy. We'll save that to last. The next thing I want to do is I want to free up these retractor muscles. So I'm going to take this, just get my probe between the retractor muscle and the body of the squid. Same thing over here. I'm going to free this up. There we go. I want you to feel that muscle. I want you to feel the muscle. And then I want you to just pull those retractor muscles back and forth. And you can see how that's actually going to move the squid's head and move his siphon. This is where the water actually exits the squid. And that water, by moving these retractor muscles, which are very powerful, the squid can actually maneuver in the water. So make sure you do that. Again, all part of dissection. The next thing we want to do is we want to remove this siphon and expose the head region. So if I turn the head just a little bit, this siphon is connected to the body of the squid via a band of tissue, and I've got my probe right underneath that band of tissue. All I have to do now is come in here and cut that band of tissue on one side, and then I'll do the same thing on the other side. Again, I'm going to come in here with my probe, and I'm going to cut that band of tissue. There we go. At this point, I'm simply going to gently peel back the siphon. I'm going to peel it back, and the only thing that should be holding this siphon onto the squid, and I'm going to go ahead and cut that, little band of tissue, is the retractor muscles. I'm going to reflect that siphon back and just kind of lay it right on the body of the squid, and now the whole head region has been exposed. I'm going to get rid of some of this latex, this plastic material. so that we can see the head region a little bit more clearly. Now, at this point, we're going to go back to the mouth. And what I want to do is I want to open up these arms, and you can see there's almost like a little space between the buccal mass, where the mouth is located, and the rest of the squid. I want to get my scissors in there, and I want to cut. And I'm going to cut again until I get to about this point right here. You can see this clear material. That's actually cartilage, and you can see this clear material. If we zoom in, you can see that sort of shiny material. And if you put your finger on it, you can feel it. That is cartilage. You need to be able to feel what cartilage feels like. That's going to be the squid's skull, and the brain's going to be right inside there. Now I'm going to go ahead and open this up, and I'm going to expose this buccal mass. So there's the buccal mass right there. Clean this up a little bit. And this is all buccal mass. Essentially, that's the oral cavity of the squid. We're going to open that up in just a minute. The next thing we're going to do is we're going to make a small incision here with the scissors. And I'm going to cut, and then I'm going to further open this up. So I'm going to open up the skull of the squid, and I'm going to gently open that up. We don't want to open it up too fast and tear tissues. And when I do that, I see this cheesy material. This white cheesy material is the actual brain of the squid. You can see it. There's a little bit more on this side. You can see it especially well right there. It's very prominent. And you can feel that right here. That's all cartilage. Get in there with your finger and feel what that cartilage feels like. You can feel it all around here. This is cartilage. That's the skull. If I open this up a little bit more, and I go to that buccal mass, there's going to be a tube coming off that buccal mass. I've exposed it. That's going to be the esophagus. If we kind of tease tissues away here gently, maybe we can free it up. So there it is. And I'm going to follow it. There it is. And if I continue up further, and I don't want to cut the ink sac, so be careful not to cut the ink sac just yet. The ink sac is up here. But if I follow it up, this is all going to be glandular material. This is the squid's digestive gland, or liver. And I'm just going to kind of cut that. And I want you to see what glandular material looks like. It's very cheesy, soft material. It's been dyed a little bit with the latex, but this is all the squid's liver. Very cheesy glandular material. You can see it right here. This is all glandular material, very cheesy. I want you to be able to recognize glandular material when you see it and feel it. We can actually follow, if we do this carefully, we can actually follow the esophagus up. So there it is. We're picking up the esophagus there. And we could literally follow it all the way up. You can see it. There it is there. And if I wanted to, I could follow it all the way up to the stomach. But let's go back to the buccal mass. I'm now going to make an incision right at the very ventral aspect of the buccal mass, and I want to expose the beak. So I'm going to get inside that mouth, make a little incision, and then using my thumbs, I'm just going to kind of open this up. And I'm going to remove that beak. So there's the beak. I'm freeing it up. I'm going to get the probe underneath that beak. And take a look at that. That's like a chisel. Squid actually uses that beak to tear food over top of that beak. So there it is. If we zoom in on it, you can see that nice sharp point. Go ahead and feel that. Just feel the tip of that, nice and sharp. The bigger the squid, the bigger the beak. And then obviously inside this buccal mass is his oral cavity, where the food goes. Make sure you take a look at that. After we've looked at all those structures, we're going to go ahead and we're going to reflect the body of the squid back. And I'm going to slowly kind of just peel it away. And I want to expose the pen. Slowly peel the squid's body away, exposing the pen. Make sure before you do this that you've gone back through and you've identified all the structures again 2-3 times, more if need be. And then and only then go back and peel the entire body away from the pen. You might have to use your scissors periodically, cut some of that material. Should pull away fairly easily. Sometimes you get a whole pen intact, and sometimes you don't. Remember, this has been preserved for months or maybe even over a year. Then what I want to do is get my probe underneath the pen and free it up. There we go. There we go. There we go. There's the pen. What I forgot to do, and we can actually do it right now, is I'm going to put the body back where it belongs, and I'm going to go ahead and cut the ink sac. See if we can get some ink out of there. Sometimes that ink is congealed, kind of solidified, and sometimes it's liquid. I'm just going to take one of the blades of the scissors, and I'm going to cut that silvery ink sac. And sometimes ink will ooze out. There it is. You can see that black ink. And you can actually take the pen, which is the squid's internal skeleton, that's his internal shell. You can actually, now in real life this ink is all liquid, but now because of the preservative it's coagulated a bit. When the squid is scared, the squid needs to retreat. It's going to squirt that ink out along with the water and create a cloud in which to escape in. So the squid uses that ink for a defensive mechanism, defensive purposes. And that's going to be it for the squid dissection.